Okay, so for this homework assignment, we're going to go over most of what we covered in cell biology. This is going to be the first one of many of the homework study guides that I'm going to give you guys so you can prepare for the semester final. So the first thing I want to review with you is the plasma membrane. And one of the major functions of the plasma membrane, why it's so important, is again, I talked about how I related it to a bouncer, how it regulates what enters and what leaves the cell, and the way that that's described is semi-permeable, and what that means is it allows some things to enter and it keeps other things out. That's important for everything. So your cell has to regulate, it has to take in nutrients, and it has to excrete waste. So you can see why that's important. And when you look at the structure of the plasma membrane, you could see that it is made out of these little funny shapes here. And if you remember that these heads is made of phosphates. And since they're made of phosphates, they tend to like water, so they face towards where there is water. So in the cell, there's water here, and there's water here. So since these phosphates like water, they are called hydrophilic. Or water-loving, okay? Now these little tails, these are made of lipids. And since they are made of lipids, they don't like water, so they try their hardest to stay away from the water, and they all face inward to stay away. And if you remember, if these heads are hydrophilic, these tails are hydrophobic. And I'm pretty sure I'm probably going to put on the final a diagram where you have to label this, the phosphate heads and the lipid tails, and label which part is hydrophilic, or which part likes water, and which part is hydrophobic, which part hates water. Okay? So, moving on, we talked about the outside layer of the cell. Next, there's two main classifications of cells. And if you remember, one is eukaryotic or eukaryote, and the other one is a prokaryote. And the main difference between the two is the eukaryote is much more complex. It has more of these little organelles in there, okay? More of these structures. And the most important one is it has a nucleus. So if you look at the nucleus in the eukaryotic cell, you can see that that's where the DNA is stored. And if you look over to the prokaryote, there is no storage space for the DNA. It's just free-flowing, okay? So the biggest differences between the prokaryote and the eukaryote is the eukaryote has that storage compartment for the DNA, and the DNA just kind of is uncoiled in there, okay? And when you look at the prokaryote, there is no storage location for the DNA, so it would just free flow, just like that, okay? And if you remember the way that I told you guys to remember the prokaryote and the eukaryote, what are they going to carry? They're always going to carry the nucleus, okay? Prokaryotes do not have a nucleus. I may put a picture on the final and tell you to identify, is that prokaryote or eukaryote? And if I do that, the major thing to look for is the nucleus. If there is a nucleus, automatically it is a eukaryote cell, okay? Then when you continue to look into the eukaryotic cell, there's more complex organelles, okay? And two of them that are commonly confused are the endoplasmic reticulum. And there are two types. There's a rough ER and there's a smooth ER. So let's differentiate between the two. Okay, so the rough ER, it is studded with ribosomes. That's why it's called rough. It has all these little spots all over them. Okay, and those spots, those ribosomes, is what makes it rough. It gives it that rough-looking texture. And if you remember what ribosomes do, ribosomes, along with the rough ER, they both serve the same function. They both do the same job. So ribosomes and rough ER both make proteins, okay? And if you remember, you are made up of proteins. Your skin, your cells use proteins for structure. Just about every single part of you almost is a protein, okay? So both the ribosomes, because there are free-floating ribosomes that aren't attached to the rough ER, the ribosomes and the rough ER are responsible for making proteins, okay? Now when we look over at the smooth endoplasmic reticulum, the smooth endoplasmic reticulum serves two main functions that are important. Number one is it removes toxins. Okay? So there's a lot of smooth ER in your liver because your liver is responsible for removing toxins from your blood. So inside of liver cells, there happens to be a lot more smooth ER than in other cells. While other cells still have smooth ER, there just happens to be a lot more in your liver. Okay? And then function number two is it makes lipids, okay? So, pretty easy to differentiate between the two. Rough ER makes proteins, and it's rough because it's studded with these ribosomes, okay? Smooth ER has no ribosomes and makes lipids and removes toxins. Now we move on to the Golgi apparatus. Golgi. And what the Golgi apparatus does is, I told you before, I gave you a pretty basic definition of it, and I described it as the post office, okay? And yeah, it is the post office. Well, what ends up happening, to look into it a little more in depth, is these proteins made from the rough ER and the lipids made from the smooth ER, they're not done yet. And what happens is when they're done, they get shipped to the Golgi apparatus. And what the Golgi apparatus does is it takes these proteins and it puts it in a package. It puts it in a vesicle. It'll modify the protein or the lipid. It'll change it a little bit. It'll put it in this package called a vesicle. And after it's done putting it in the vesicle, then it's going to ship that wherever it needs to go. Could go to another cell, could go stay inside of that cell. But the Golgi apparatus receives proteins and lipids, it modifies them, and then it ships them off to where they're supposed to go. It ships them off to their destination. So that's why it is called or described as the post office of the cell. Okay? Next, we're going to talk about the mitochondria. And if you remember the way that I told you guys before, how to remember what the mitochondria does, the mitochondria is described as the powerhouse. The powerhouse of the cell. And a little bit later on, we talked about respiration. And respiration is the process used by your cell, specifically by your mitochondria. And in that process, it takes the food that you eat, which the food that you eat gets broken down into glucose. And when it takes that glucose, it converts that glucose into the energy molecule. And if you remember, that is ATP. Okay? So the mitochondria's function, the purpose, is to take food that you eat after it's broken down into glucose, and it converts that glucose into ATP. And that's why it is called the powerhouse, because all of your cellular functions run off ATP. If you don't have a lot of mitochondria or any mitochondria, you're not going to get a lot of the ATP. Okay? So again, the mitochondria, powerhouse of the cell, and it makes ATP, okay, from glucose. makes ATP from glucose. Okay? And next we're going to go over the plant cell. The plant cell is eukaryotic. How do you know that it's eukaryotic? Well, it does have a nucleus. It has a nucleus right there. That's where the DNA is stored. So the plant cell has every structure that an animal cell has, okay? It has everything that an animal cell has plus 3 additional structures, okay? 3 of them. Number 1, let me go ahead and erase that number 3 real quick. Number 1 is a cell wall, okay? And the cell wall is made of cellulose. And all cellulose is it's a polymer, and it's a bunch of repeated links of glucose molecules, okay? So the cell wall is the outer layer of the plant cell, and that's what gives it its structure, because plants don't have bones. They don't have a skeleton like we do. So what holds them upright is the cellulose. That's what helps give them their structure, okay? Structure number 2 that plants have that we do not is a large central vacuole. Okay, and that's this big guy right here. What happens for the large central vacuole is this is the storage for water for the plant cell. Okay, so when a plant is hydrated, what's going to happen is this large central vacuole is going to be filled with water, and it's actually going to push, it's going to expand, and it's going to push all the organelles up against the cell wall. So it will get some pressure, it will get some structure from the large central vacuole when it has a lot of water in it, okay? So both the cell wall and the central vacuole assist in the plant structure. And structure number 3 that plant cells have that we do not is the chloroplast, okay? And if you remember, this is the chloroplast right here, these green things. The chloroplast is the site, okay? Put it in green. It is the site of photosynthesis. And in another homework assignment, we'll review the process of photosynthesis, okay? But the chloroplast is the site of photosynthesis, okay? Okay, so that was it for the organelles and the cell structures. Now I want to review cell transport, okay? And if you recall, there are 2 types of cell transport. Number 1 is passive transport. And if you remember, passive does not require energy. And the energy that we use is ATP, okay? So, no energy. We don't need it. It just happens. And the first type of passive transport is diffusion. And if you remember, diffusion is the movement of molecules from high concentration. So if you look at this picture right here, you have high concentration of molecules, and it's going to move from high to low, okay? So again, you have a high concentration of molecules here, you have none over here. And these molecules are going to diffuse from high concentration to low concentration. They're going to go where there is no molecules, okay? And they're going to continue to move until it reaches a state of equilibrium. And what equilibrium means is there's an equal amount throughout all areas, okay? That's diffusion, and that is one form of passive transport. Another form of passive transport is osmosis. And the definition of osmosis, put this in blue, The definition of osmosis is the movement of water across a cell membrane. Okay, so if you remember, the membrane is what regulates what enters and what leaves. So one of the things that can freely move across a cell membrane is water. Okay, and how do you remember? I'm going to go ahead and erase this real quick. How do you remember where water is going to go? Well, if you identify where a majority of the solutes are, so a solute could be anything. A solute is something that dissolves in water. And if you look in these pictures and you identify where the majority of the solutes are, that's where water is going to go. Okay, so water, remember, water follows solutes. Okay, so if I look in this first picture here, in this first picture there is it is a isotonic solution. And if you remember, in an isotonic solution, the amount of solutes outside the cell and the amount of solutes inside the cell are equal. Okay, they're the same. So the amount of water that travels inside of the cell is the same as the amount of water that travels outside the cell. Okay, and if we look at number 2, it is a hypertonic solution. And remember, hyper means a lot. So anytime you hear hyper, that means in excess. So what are we talking about? Well, we're talking about the solutes. Okay, so again, there's a lot of these solutes outside of the cell. And again, what does water follow? Water follows solutes. So since there's more solutes outside of the cell in a hypertonic solution, the cell is going to shrink. Okay, it's going to shrivel. It's not going to be good for the cell. Okay, and then third, if we look at a hypotonic solution, if you remember, hypo means low. There's not a lot of solutes. Okay, so in a hypotonic solution, there's actually, since there's less solutes outside of the cell, what does water follow? Let me go ahead and put some more solutes inside of the cell for you. That's a better representation of a hypotonic solution. Okay, so there's a lot of solutes inside of the cell. There's not a lot outside. So again, what happens? The water is going to travel inward. Okay, and that cell is going to expand and potentially pop. It could potentially burst. Okay, from so much water entering the cell. And the final thing we're going to cover in this is active transport. So, real quick, osmosis is passive. So, no ATP. No energy is required in osmosis. Same as diffusion. No ATP, no energy. No energy in osmosis. Active transport, since it is active, yes, ATP. You must have energy, okay? And in class, we covered 3 forms of active transport. I only want to review just these 2. So the first one is exocytosis. And if you remember, exocytosis is when the cell is, it reminds me, exo sounds like exit. So exocytosis is when the cell is taking something out, okay? So it has waste products and it's trying to get rid of it. Now when we look at endocytosis, endo means into, okay? So in endocytosis, something is traveling into the cell. Could be anything, proteins, could be nutrients, okay? Probably something good for the cell. So something is coming in. And if you remember, the important part to remember for exo and endocytosis is it uses a vesicle. And a vesicle is the same package that the Golgi Apparatus makes, and it is also made of a phospholipid bilayer. Okay, it's made out of the same type of membrane that the cell that surrounds the cell. Okay, and I'm gonna go ahead and call that it for this lecture. And this will be number 1 of a few more to go. I promise you guys, if you complete the homework assignment, if you review the study guide just a few minutes a day, after this, you have your study guide, I'll give you back your homework. Just look at it a couple minutes a day, and I promise you for the final, it will be a cakewalk, okay? And actually, one more thing, too, for those of you guys that may have been wondering, if you don't remember, let me go back real quick. Back to prokaryotes and eukaryotes. In case you were wondering, I gave you a bunch of examples of what a eukaryote looks like. So you might be asking, what is a prokaryote? A prokaryote is bacteria. They're not nearly as complex as eukaryotic cells or animal cells. They're not as complex. They're very simple, and they're usually single cellular. And next semester, we're going to go over a lot more about bacteria. We might even get to culture some, grow some. So it should be fun. And again, hope you guys enjoyed it. And make sure you do your homework. Bye.